So I'm going to tell you a story about elevated nitrous oxide production outside of anoxic zones in the ocean, and I'll define what I mean by elevated nitrous oxide production. This is still a little bit of a work in progress, and I'll try to be careful at pointing out where the story is still a little incomplete or half-baked, perhaps. So this is a story that pulls together new data and 3 different modeling approaches, and so it involves a lot of collaborators. Those are Bonnie Chang, David Nicholson, and Colette Kelly, who are contributing nitrous oxide data from the GoShip program. My friend and frequent collaborator, Daniele Bianchi at UCLA, who I work on modeling projects with, and his graduate student, Daniel Clements. My clicker is not working. Okay. The computer is unresponsive to any clicking. Maybe I'll try to X out of this and okay. Should I just try unplugging and back in? This has never happened on any other. I'm gonna try unplugging. Yeah, and this was just working as I was testing before. Sorry, everybody.
Do you need a pointer?
No, I have that here. And now I feel like there must be an issue with our system here, because now it's not picking me back up.
I have to do without slides. It happened to me once.
Okay. Well, so this screen had gone dead. I don't know if that. But now the question is whether I need to sign back into our. I'm on Teams. Okay. I should be still. Okay. Can we move on? Let's pick up where we were. So I'm telling you this story. Here's the outline of the story I'm going to tell you. So I'll start off with some background where I'll just talk quickly about the climate significance of nitrous oxide and review what the marine sources are and how oxygen affects those sources, and review our 2 different perspectives on N2O production in the ocean, what we learned from biological measurements and geochemical approaches. And I'll spend a fair amount of time on those 2 background points, because I would rather bring you all along and help you understand the problem I'm trying to solve than get through all of my results. So after that, I'll talk about our new geochemical approach for estimating the nitrous oxide yield. Again, I'm going to define exactly what we mean by the nitrous oxide yield. Our new approach involves machine learning and diagnostic modeling. And ultimately, as a spoiler alert, the main result I'm going to show you is that there's more nitrous oxide production happening in low oxygen but not fully anoxic waters than we would expect from what we know biologically. And then I'm going to try and get into the reasons why that might be happening. And the 2 mechanisms I'll look at are physical variability in the ocean and then particle microenvironments. And then at the end, I'll try to come back to big picture climate stuff by just saying a couple of things about the future of nitrous oxide sources in the ocean. So into the background, as we're all probably aware, humans are emitting nitrous oxide to the atmosphere, largely through agricultural processes, also through the fossil fuel industry. And so nitrous oxide levels have been increasing in the atmosphere from their pre-industrial level of 270 parts per billion. And now by 2024, the concentration has risen by about 20% above that pre-industrial level. The bottom plot down here shows us that the total radiative climate forcing of all the extra nitrous oxide that's accumulated in the atmosphere is about 0.2 watts per meter squared. And when we compare that to the radiative forcing from other greenhouse gases, CO2 and methane, it works out to about 10% of the total radiative forcing we're imposing on the climate system through greenhouse gases is due to our nitrous oxide emissions. So very important from a climate standpoint. So if we want to look into the climate future, we certainly need to know something about the future of human N2O emissions, but we also need to be worried about how climate change and how it impacts the environments could impact the natural sources of N2O from the atmosphere. So natural sources to the atmosphere. So jumping over to this side of the plot, the two main natural sources are from wetlands and soils on land, and then from the ocean. So oceanic emissions around 4.7 teragrams per year, similar order of magnitude to all of the anthropogenic emissions. So if the way that we're perturbing the climate perturbs the marine source of N2O to the atmosphere, we should worry about the climate feedbacks that could induce. So there's our motivation for understanding nitrous oxide emissions from the ocean. And I just want to point out at this point the most recent estimate of these marine nitrous oxide emissions incorporated into the Global Carbon Project budget are from this paper that I was a co-author on, Yang 2020, that used a machine learning approach to estimate the nitrous oxide emissions. And I only mention that because we're going to see a similar approach used later in my talk. Okay, so if we're worried about nitrous oxide emissions from the ocean and how they respond to climate, we need to understand what are the actual mechanisms producing this N2O in the ocean. So for that, we'll go to a simple picture of the nitrogen cycle in the ocean, and we'll start by seeing how this looks in well-oxygenated regions of the ocean. So in the well-oxygenated ocean, all of our fixed nitrogen species, ammonium, nitrate, nitrate, are all taken up by phytoplankton to produce organic matter in the euphotic zone. That organic matter decomposes, remineralizes back to ammonium, and in the surface ocean, there's rapid recycling between organic matter and ammonium. But below the euphotic zone, that ammonium is nitrified back to nitrate through the intermediate of nitrite. Okay. So where does N2O, nitrous oxide, fit into all of this? It fits in because this first step of nitrification, conversion from ammonium to nitrite, is not perfectly efficient. Actually, it turns out that during that process, a small fraction of all the nitrogen atoms end up in the form of N2O instead of nitrate. And it's a small fraction. It usually works out that around 0.3% as much N2O accumulates as nitrate accumulates through this nitrification process. So this source of N2O is pretty ubiquitous through the ocean. We don't fully understand the reasons why some of the ammonium is converted to nitrous oxide, but we know it happens. And once the nitrous oxide is produced, there's no real sinks for that in the oxygenated ocean. So eventually this will reach the surface and outgas to the atmosphere. So there's our picture of the oxygenated ocean. Of course, we know not all of the ocean is well oxygenated. Right? You will recall from your chemical oceanography classes that if we think about the oxygen distribution in the ocean interior, we can think about parcels of water at equilibrium with the atmosphere, leaving the surface and gradually losing oxygen along their transport pathway due to the remineralization of organic matter. So wherever in the ocean the most integrated remineralization has occurred following water along its pathway, that's where we'll find the lowest oxygen concentrations.
Are you still there? I just want to borrow your headset.
Somebody's talking to me through this Teams meeting. Should I ignore it? Assumes nobody's trying to ask me a question. Okay. So find those lowest oxygen concentrations in the tropical ocean, where in the thermocline depths of the tropical ocean. I'm showing you pictures from 400 meters here. And in these regions, the waters have spent a long time out of contact with the atmosphere and have been exposed to high remineralization rates relatively shallow in the water column. So we find our lowest oxygen concentrations in the tropical thermocline. And in specific regions, most notably here in the eastern tropical Pacific, oxygen gets really low all the way down to disappearing completely in what we call the oxygen minimum zones, or suboxic zones of the ocean. So we find suboxic zones in the eastern tropical North Pacific off Mexico, in the eastern tropical South Pacific off Peru, and in the Arabian Sea in the Indian Ocean here. So that's where we get all of the way to suboxic or even fully anoxic conditions. Okay, so how does the nitrogen cycle change across this oxic to anoxic gradient? The first thing that happens that matters for nitrous oxide is this production of nitrous oxide through nitrification increases, by which I mean that the yield of nitrous oxide per mole of nitrogen nitrified increases from this 0.03% up to larger fractions. So I should point out that at this stage, my talk was called Elevated Nitrous Oxide Production. When I say elevated nitrous oxide production, we're talking about nitrous oxide production above what we would expect from the oxic nitrification yield. So we're going to see two mechanisms that can lead to this elevated production. The first is just that this yield during nitrification increases from the 0.03% up to into the range of 2% or even higher. And we know this from a number of studies that have done incubation experiments, either in the lab or in situ at sea, and have measured the yield of nitrous oxide production per mole of nitrate produced during nitrification. Okay. Before moving on from this plot, I just want to point out that in some of the upcoming plots, we're going to see this same quantity, the yield of nitrous oxide produced during nitrification, but we'll see it in a slightly different way, which is a yield of N2O per mole of oxygen consumed during the organic matter remineralization. And that's just a stoichiometric conversion using the stoichiometry of remineralization. But that's just to make you aware that in some of the plots, we'll see this coordinate system change, and the numbers change by about a factor of 10 when you look at nitrous oxide per oxygen consumed. Okay, so there we have our first mechanism for elevated nitrous oxide production. And this, according to the lab experiments, the incubation experiments, if you fit a line through that, this elevated yield seems to occur at suboxic conditions, oxygen below 5 micromolar concentrations. Okay, my diagram has all of a sudden got more complicated to represent the additional nitrogen cycle pathways that occur once we reach suboxic or anoxic conditions. And for our story, the important extra pathway that's been added is the denitrification pathway. This is when there's no oxygen available to remineralize organic matter. Microbes will use nitrate as an alternative electron acceptor and reduce the nitrate to nitrite. And they'll use nitrite as an electron acceptor and reduce that to nitrous oxide, and further reduce that all the way to dinitrogen. Okay. So in fully suboxic water, we have this whole extra mechanism that can produce N2O at elevated levels beyond what we'd expect from the usual nitrification yield. And again, from incubation experiments, we know the oxygen concentrations that this process should happen at. So what I'm showing on the right here are some incubation results that show the degree to which this process of N2O production during denitrification, the degree to which that becomes inhibited by oxygen availability. And what they show is that at oxygen concentrations above 1 micromolar, 1,000 nanomolar, that process is almost completely inhibited. So this production pathway should really only be happening below micromolar oxygen levels. And then the top plot is showing you the same thing, the inhibition, but now for the reduction of nitrous oxide to dinitrogen. And that process becomes inhibited at even lower oxygen concentrations, around 0.5 micromolar. So this should really only be happening in very anoxic waters. This means there's kind of a narrow oxygen window somewhere between 1 micromolar, which inhibits this process, and 0.5 micromolar that inhibits this process. There's that narrow oxygen window where we can get major accumulation and net source of nitrous oxide without it immediately just being reduced all the way to N2. Okay, so what we've seen so far gives us a pretty simple picture, which is that elevated N2O production above what we expect from normal nitrification should only be happening in very low oxygen waters, right? We had the elevated nitrification yield at concentrations below 5 micromolar, and now we have this whole extra pathway which should be happening below 1 micromolar oxygen. So this should be confined, this elevated production should be confined to very low oxygen waters. That's great. That would give us a nice way to predict how nitrous oxide production could change in a future climate. But of course, things are not that simple, because there's a whole other way of looking at trying to estimate nitrous oxide production that gives a very different story. And this is the method of looking at large-scale distribution of traces to kind of parse out process rates. Right? So this is what we would call a geochemical perspective on nitrous oxide production. So I'll walk you through a couple of plots to show the type of inferences people have drawn from the large-scale distribution of N2O in the ocean. What we're seeing on the first plot here is just a bunch of data scattered that is the disequilibrium of nitrous oxide, so above what you expect from atmospheric equilibrium on our y-axis, and apparent oxygen utilization on our x-axis, right? So as a water parcel moves away from the surface, oxygen is going to get consumed and nitrous oxide will get produced through our nitrification process. And following the normal oxygenated yield, right, we would expect the points to follow a slope on this plot illustrated by the purple arrow, right, following our normal nitrous oxide yield with a stoichiometric conversion to convert to oxygen units rather than a nitrogen yield. We expect our points to plot along a slope like that. But what this plot shows, while a lot of the data does do that at relatively low oxygen consumption, so high oxygen concentrations over here, right, as we go towards high AOU, so low oxygen, a bunch of the data starts plotting well above the line. Right? The next thing we'd want to know is what actual oxygen concentrations is this elevated N2O production actually occurring. And in order to figure that out, you have to plot the data in a slightly different way. So this is a geochemical approach first used by Nevison, 2003, where on our y-axis, you now plot the ratio of excess nitrous oxide over AOU. And on the x-axis, you plot the oxygen concentration, and you just plot data points that lie along a single isopycnal. So now we can kind of interpret this as a water parcel moving along an isopycnal from high oxygen concentrations, having the oxygen consumed, and having both nitrous oxide produced and oxygen get consumed, AOU. So on a plot like this, if we're just following the regular well-oxygenated N2O yield, that would plot as just a straight line like this, right? Because if you produce nitrous oxide at a constant ratio to oxygen consumption, right, then that doesn't necessarily change the concentration ratio in your water parcels. So we would just move along the x-axis like this. And now in this way of looking at the data, it kind of tells us that a lot of the data seems to do that at high oxygen concentrations, but once we reach oxygen concentrations around 100 micromolar or so, the ratio of nitrous oxide to AOU increases in the water parcel, telling us that the yield of N2O production must be higher than we'd expect from that, from the well-oxygenated, the canonical well-oxygenated value. Okay. In order to compare exactly what we're learning from this plot to this plot, you have to go through a few more steps that they tell you in Nevison. So on this plot here, we're still looking at total accumulated nitrous oxide and total accumulated AOU along the entire transport pathway of a water parcel. To compare that to the biological rates, we'd want to convert to figure out what is the instantaneous yield at any particular time of N2O production to oxygen consumption. And you can get that by taking a derivative of this curve here. And the ultimate result of that is that the geochemical approach based on the large-scale distribution of N2O would tell you that the nitrous oxide yield is just higher than what we'd expect from biological incubations at oxygen concentrations all the way below 100 micromolar. So in this case, the distributions are telling us that the nitrous oxide yield starts ramping up at relatively high oxygen concentrations, certainly not just as we enter the very suboxic or anoxic waters. Yeah. And in the study, the way that people largely interpret this is that the data is telling you there must be a higher yield of nitrous oxide during the nitrification process, because the only other option for this extra N2O production would be that you have denitrification, that second source, happening at oxygen concentrations up to 100 micromolar, which is really hard to reconcile with how we understand that process. So we have this big disagreement between what we learn from incubations and what we learn from large-scale distributions. And that's a problem because ideally we want to be able to predict what happens to N2O production in the future ocean, right? And if we have a very unclear picture of how N2O production responds to oxygen, we have no clear way of modeling and therefore predicting nitrous oxide production. So how do we explain this? Well, we could explain it either by saying the biological rate measurements are wrong, right? Maybe the microbes in incubation just act differently than they would in the natural environment, or we can point to some potential issues in our geochemical methods. It's based on relatively sparse data. There are potential mixing biases, because to convert to a curve on this plot, you also have to make some assumptions about end member mixing. So there's potential issues in the geochemical method too. And what I'm going to do going forward is to present a new way at coming at the geochemical approach that hopefully bypasses those issues. But before I do, I want to point out why this is especially important as we start thinking about climate predictions. So it should come as no surprise to any of you that the ocean is already deoxygenating, and we expect it to deoxygenate further into the future. So this is output from the most recent generation of climate biogeochemical models. And the red curve here is showing us our what was traditionally known as the RCP 8.5 business as usual scenario, right? So the red curve is showing us our business as usual scenario, and under that scenario, by the year 2100, the ocean loses somewhere around 16 millimoles per meter cubed of oxygen on average. across the ocean. Right? And that's due to a combination of warming, reducing the saturation of oxygen in the ocean, and a slowdown of the ocean circulation, which ventilates oxygen into the ocean interior. But the actual pattern of oxygen loss in the ocean is much more complicated and even unclear. So on the right-hand side here, I'm showing you a plot at 400 meters. Again, that critical depth, showing the change in oxygen concentration by the year 2100 in these climate models. The brownish colors are showing us that in much of the ocean, as we'd expect from this curve, the ocean is losing oxygen. The little dots all over the map show you the regions where all of the different climate models, and about 17 of them were used in this paper. The dots are showing you where all the climate models are in pretty good agreement about what's going to happen. Right? The ocean is going to lose oxygen. But you'll notice as we get into the tropics, the currently low oxygen regions of the ocean, we have no dots. The models don't agree with each other. And actually, if we just take an average across all of the models, which is what we're plotting here, they actually predict there's going to be a slight increase in the oxygen content of the tropical ocean. And that's because most of the models predict that tropical upwelling will slow down in a warmer climate, and so there's less organic matter production and export to consume oxygen. And now it becomes clear why our quandary from before is important, right? The future of tropical oxygen minimum zones is unclear, but most models predict that they'll actually shrink, right, as the ocean gains oxygen in that region. So now if elevated nitrous oxide production is confined to really low levels of oxygen, right, like the biological data suggests, then the marine nitrous oxide source is actually likely to decline in a future ocean as the OMZs contract. But if, like the geochemical tracer data would suggest, if nitrous oxide production is more prevalent, even at higher oxygen concentrations, then the marine N2O source could increase in a future ocean. So currently we don't even know which direction N2O is likely to move, and N2O production will move in the future. Okay. So that's why we need to resolve the problem. Okay. Going into my methods, I've been told that this is not a big modeling department in general. So the first thing I'm going to do is kind of make some definitions of different types of models, kind of the three classes of models that are widely used in the biogeochemical modeling field, just to help you understand what they are. And then I'll show you how I'm actually using all three of these in my work. So the first definition we could make is a prognostic model. That's a model that explicitly is representing processes based on our best understanding of how those processes work, guided by observations, right? So this is a process-based model that you can use to make a prediction. A second modeling type would be a diagnostic model. This is a model where our goal is to extract process information from data, from tracer observations, right? Not by resolving the processes, but often just by solving budget equations in an ocean circulation model to figure out what must be the net source or sinks of traces we're interested in. And then a final class of model would be machine learning models, kind of a trendy method at the moment. You can, for our purposes, think of a machine learning model as essentially just a fancy nonlinear statistical model whose goal is to predict some unknown property as a statistical function of a bunch of well-known properties, right? So this is three categories of model. And what I'm going to do here is first walk through how we use these two types of model, machine learning and diagnostic modeling, to come up with a new estimate of the nitrous oxide yield. Right? And then in moving on from that, I'm going to use a couple of different process-based prognostic models to try and understand the mechanisms that could result in the results I'm going to show you from here. Okay. So first, our method for attempting to reconstruct what nitrous oxide yields look like in the ocean geochemically. So the starting point for this work is a big data compilation of all the N2O measurements we can find in the ocean that is built upon a data compilation by Nevison. And then for the 2020 paper that I talked about before, we added in all of the data that's available in the Memento archive and additional data from the GoShip program provided by our collaborators. And then for this work here, we've added in some even newer GoShip data as well to form a big data compilation of nitrous oxide. If we want to then diagnose sources and sinks of nitrous oxide, the first thing we need to do is fill in all of the missing gaps on this map, try and get to kind of a globally continuous climatology of nitrous oxide, and that's where machine learning comes in handy. So the next step is that we use a particular type of machine learning model called a regression forest that just is trained to predict the nitrous oxide concentration as a statistical function of many other physical and biogeochemical properties, so temperature, salinity, oxygen, nutrients, all of these things, all things that we have but much better mapped products for than we currently have for N2O, right? So if you can make a statistical function for N2O as a function of things whose global distributions you know very well, then you can come up with a global prediction for the N2O distribution. And that's what we get through our machine learning. And I'm showing you the nitrous oxide distribution at 400 meters here that we get from this approach. And then just a plot to show you that when you run these machine learning models, you save back some of the data and don't use it to train the model. So you have an independent data set that you can use to assess how well the model did. And this plot down here is showing you that our model does our machine learning model does a pretty good job of reproducing data that it wasn't trained using. Okay, so now we have an estimate of the global distribution of nitrous oxide. Now we can go on to try and estimate sources and sinks, move into the diagnostic modeling component of our work. So the way that works is that you start with a budget equation, a conservation equation for nitrous oxide. Just assume that time rate of change of nitrous oxide has these two components, some net biological source or sink, and then the circulation terms, advection and diffusion. Right? And then we assume steady state. So now a classic budget calculation where we just assume a balance between sources and sinks. So now what we have is just our net source or sink of nitrous oxide must equal all of the circulation terms. And those circulation terms can be calculated in an ocean circulation model. In our case, we use the observationally constrained OSIM circulation model. And you just solve for all of the physical sources and sinks of nitrous oxide. And then we have to assume that's equal to net biogeochemical sources and sinks. So that now gives us the map on the right, plotting out between 200 and 500 meters on net source or sink of nitrous oxide. And you can clearly see a few things here, right in the cores of the OMZs. We have a net sink of nitrous oxide, presumably due to that final step of denitrification, converting it to dinitrogen. And then through the tropics and other regions of the ocean, we have major net source of nitrous oxide. To have something we can compare to the yield plots we've been looking at before, we need to do a separate budget calculation for oxygen, right? Remember, we've just been looking at the yield of nitrous oxide production to oxygen consumed. So we need to use the same approach to come up with an oxygen utilization estimate. So solving the exact same budget calculation for oxygen, we can come up with an estimate of the oxygen utilization rate, and now bring us to this plot here. So this is the main result that I want to show you. And I'll talk through it and then compare to some of the previous views of the nitrous oxide yield we had before. So all I've done to get this plot is take my diagnosed net source or sink of nitrous oxide divided by the oxygen utilization rate, and then we have a yield of nitrous oxide production versus oxygen utilization. Here, because this is a diagnostic model, we can't parse out N2O produced by our different pathways, nitrification or denitrification. We're just looking at a net source. So right now we have no real process information, but we can look at how that N2O yield varies in our model as a function of oxygen. And that my big red cloud in the background here is just all of the data from this model. So every grid cell in our model, obviously you can see there's a lot of scatter in there. I've turned it into a relative frequency plot. And then to try and pull out some general patterns, I've done some binning into different oxygen bins, so we can just get a sense of how this yield tends to vary as a function of oxygen. So my black dots are the average yield in 1 micromolar oxygen bins, and the blue line is our median yield. And we can see a few interesting patterns here that are best described by taking our blue line and now comparing it over to our two previous views of the N2O yield. So the red curve is that yield we get from the incubation experiments, our biological view. The green line is what we get from the previous geochemical approach, and now the blue line is our new reconstructed yield. And you can see that relative to what we expect biologically, there's elevated yield, actually all the way up to the highest oxygen concentrations we see in the ocean. And then especially the yield starts to ramp up at oxygen concentrations between 100 and 150 micromolar of oxygen, in reasonably good agreement with this previous geochemical approach. So it seems like the results from that previous geochemical approach were not just a spurious consequence of using sparse data or using relatively simple n-member mixing models. It really does look like in this low oxygen water, but not fully anoxic water, there's much more N2O production than we would expect from the oxic nitrification yield. Okay. And then just in case you're wondering, at the very low oxygen levels, my blue curve dips all the way down to negative yields. This is where we're just picking up the net sink of nitrous oxide in the core of the oxygen minimum zones, right? So the last step of denitrification, consuming N2O. Okay, so now we just have a couple of options. We have to either declare that the biological incubation experiments are just wrong for whatever reason, or we have to try and find a way to reconcile these two views together, right? And we should at least try to reconcile them before we declare that the incubation experiments are wrong. So the only mechanism we can really imagine to reconcile these yields is oxygen heterogeneity. So just a quick thought experiment. In our geochemical approach, we're working with a coarse resolution model here, big grid cells, 1 by 1 degree latitude and longitude, and we've taken a purely steady state approach, right? So we're estimating time mean N2O production, time mean oxygen consumption, and then we're plotting that yield as a function of time mean oxygen concentration. But imagine in the real ocean, one of our big grid cells actually has small patches that are anoxic, or for short periods of time, it becomes anoxic. Then there could be N2O produced in that grid cell, which our method would pick up. And our method would say that that N2O production is happening at high oxygen because the time mean climatological oxygen is high. So oxygen heterogeneity could be a way to reconcile the views. And there's two specific hypotheses about heterogeneity that we could explore. The first is that physical heterogeneity, just due to the kind of turbulent nature of these oxygen minimum zones, drives ephemeral N2O production in regions that have high climatological oxygen, right? Just to give you a quick glimpse at how temporally variable oxygen can be around these low oxygen regions. What I'm plotting here is going into the World Ocean Atlas, into the raw data, and just plotting out the time mean oxygen in the Eastern Tropical Pacific. And then on the right here, I'm plotting the standard deviation of all of the data from each grid cell. And you see that there's a band right around the margins of the oxygen minimum zone where the standard deviation in oxygen is of similar magnitude to the oxygen concentrations themselves. That means these are regions where oxygen is really just bouncing around between 0 and very high values. So these are regions where, even though we have relatively high time mean oxygen, they could be temporarily anoxic. And this is just a second. I'm going to just skip this actually in the interests of time. So I want to go on now to talk about a tool that we decided to use to look at this relationship between time mean oxygen, instantaneous oxygen, and N2O production. And that tool, now going on to our next modeling category, prognostic models. That tool is this model that Daniele Bianchi has been developing at UCLA, which takes a high resolution ROMS model of the Eastern Tropical South Pacific region and builds inside it a biogeochemical model that contains a very detailed representation of the nitrogen cycle, right? So this is a very high resolution model that, as you can see here, is predicting that all kinds of little, very low oxygen anoxic eddies are constantly spinning off from the OMZ. And of course, our hypothesis is that N2O production in these anoxic eddies could be fooling us into thinking that N2O production is happening at high oxygen levels. Okay. So she builds inside this model, a detailed representation of the nitrogen cycle. If I were to show you the N2O production rates, they would be just as patchy and variable as the oxygen concentrations itself. For our purposes, what we're really interested in is how do the time mean N2O production now line up with the time mean structure of the OMZ. So what I'm going to do is show you some time averaged results now across this line here, 10 degrees south. On the left-hand plot here, we have the nitrous oxide concentration itself. And our contours here are showing us the structure of the oxygen minimum zone, the 10 micromolar contour and the 1 micromolar contour. What I should point out before I go into that figure in too much detail is that in Daniele's model, all of these processes are parameterized using the incubation experiments, right? So these N2O production pathways really are only going to be happening at very, very low oxygen. So in the central panel here, I'm now showing you the net source or sink of nitrous oxide due to our denitrification pathways. And then on the right here, the source due to the nitrification pathway. And the quick walk through the results here are that the middle plot is showing us that we have a large net sink of nitrous oxide due to denitrification in waters where you wouldn't expect it to be happening based on how the process is hard-coded into the model. Right? Remember this, the model is actually resolving the production process only happening at sub-micromolar oxygen levels, but when you just take a time average view, it looks like nitrous oxide is being produced in this whole shell with oxygen levels up to 10 micromolar and even outside that shell as well. And that must be the impact of these anoxic eddies, which don't show up in the time mean oxygen concentration, driving this process outside of the OMZ. And then nitrification. Nitrification reaches its peak right below the euphotic zone, but probably can't be seen due to the color scale here, but nitrification reaches quite high levels right above the oxygen minimum zone here. So a better way of looking at this data that's going to be more comparable to what we've seen before is to just take all of this, these net source and sink rates, divide by our oxygen consumption rate, so we can again look at one of these yield curves, right? So we take all of that data from the high resolution model and look at it in the yield way of looking at things, exactly as I did from the diagnostic model. So the important things to point out here is that, again, in the model, nitrification is following this green yield curve from the incubation experiments, right? And denitrification is only happening at oxygen concentrations below 1 micromolar. But when we look at things time averaged, it's showing us that it seems like there's high nitrous oxide yield at two different bands of high oxygen. Right? So all the way down here on the right, we have high nitrous oxide yield occurring at very high oxygen, 200 to 250 micromolar. Looking deeper into the model, we can figure out exactly what that is, right? This is elevated nitrous oxide yield during nitrification caused by heaving of the upper oxycline, right? So all of this physical variability sometimes causes the upper oxycline to heave towards the surface, and now nitrification occurs, for some short period of time at very low oxygen, and it drives a higher yield than you'd expect from the time mean oxygen. Right? So this hump here is due to nitrification. On the
could you just I'm sorry, like
yeah.
What's it like the main plot, the boxes?
Oh yeah, so right. So this is taking the same view that I showed from the diagnostic model, but it's just taking our our high resolution model results. This is the ROMS model results, and it's forming a yield from the nitrous oxide production rates and the oxygen consumption rates. It's then binning them onto a coarse grid and then plotting just the time averaged view, right?
Okay, those are wrong. That's what
Right. So this is the ROMS data just turned into a yield curve. Right. So the little rectangles are just showing again the relative frequency of where the yield falls, right? So kind of taking all the pixels in our model. And so the elevated yield at very high oxygen, we can pin that down to nitrification. The elevated yield, beneath 50 micromolar oxygen, we can attribute that to denitrification in the model. So this is, like I said before, the anoxic eddies spinning off from the OMZ core, N2O production occurring in the anoxic eddies and driving elevated yield at relatively high climatological oxygen. So our first hypothesis was that maybe our new reconstructed nitrous oxide yield could be explained by this physical heterogeneity. We can now actually compare what on our blue curve we can actually explain through the physical heterogeneity. So going back here and just pulling kind of an average line through all of the boxes that we just looked at. So kind of the average yield curve from ROMS. We can plot that on top of what we had before and show that this is a good way to explain a couple of the features we saw in our diagnostic model result. It explains our high yield at very high oxygen due to nitrification. It explains the elevation below 50 micromolar, but critically, it doesn't explain any of this intermediate period portion of the oxygen spectrum here, right? Between 50 and 150 micromolar, we still have way more N2O production according to our diagnostic result, way more N2O production occurring there than we can explain through biology and physical heterogeneity, right? Or from our incubation results and our physical heterogeneity. So now we come up with a second hypothesis that I'm just gonna talk about very quickly because we lost some time due to technical issues, and I want to make sure we have question time. But second hypothesis is that a second way of creating oxygen heterogeneity is inside of particles, right? So another hypothesis would be that localized denitrification inside anoxic particles could be driving N2O production at high ambient oxygen. So let me just talk you through this idea quickly. We've long known from theoretical calculations and lab experiments that anoxic zones can form inside of large, fluffy aggregate particles just due to high localized respiration rates occurring inside those particles and the limits on how quickly oxygen can diffuse into the particles. So this lab work showed that particles can become anoxic inside, even at very high ambient oxygen concentrations. And all kinds of interesting observations from the field have recently been attributed to microenvironments inside particles like this, often without a lot of supporting data, it has to be said. But it's a convenient place often to reconcile confusing observations like sulfate reduction products being found in waters where there's way too much oxygen to really allow sulfate reduction to occur. And the reason we got really interested in this hypothesis was a paper from just last year showing the mechanism in work in coastal waters in the East China Sea, right? So they had 3 study regions here, regions where there's very elevated nitrous oxide levels in the surface water, and they did isotopic labeling experiments to find out where that N2O was coming from. And confusingly, they found that most of it must be coming from denitrification rather than nitrification, which is really weird in oxic coastal waters. And then through doing some filtration and repeating the experiments with and without particles, they could attribute this nitrous oxide production to denitrification specifically occurring inside particles. So this was the first time that we've really had good evidence for N2O production in oxic waters from these in these particle microenvironments. How long do I have? Should I wrap it up in 1 minute? Couple minutes? Okay. So luckily, again, we have a tool to test how this mechanism may contribute to our results. And that tool is a model that Daniele, my collaborator, and I developed a few years ago, in which we actually attempted to resolve the chemical microenvironment of particles in a model. So we simulate a size spectrum of particles as they sink and remineralize and disaggregate through the water column. And then in every single particle, we solve for its internal chemistry by first assuming that oxygen gets consumed by respiration, and then nitrate gets consumed, and then sulfate gets consumed. So we resolve the microenvironment structure of the particle. And the main result of that study was that with this model constrained by the best available data we have, we estimated that even in relatively well oxygenated waters, a pretty high fraction of total remineralization could be fueled by denitrification rather than aerobic respiration. So on this map here, the green line is a 100 micromolar oxygen line. And even at oxygen concentrations going up to 100 micromolar, you see somewhere between 0.1 and 10% of total remineralization could be fueled by denitrification rather than aerobic respiration. And if particle-bound denitrification is really that widespread, then N2O leakage from these particles could really be that widespread as well. So just the final result I'll show you is that we extended this model now to allow for some fraction of the nitrogen consumed by denitrification inside the particle. We allow some fraction of this denitrification to be incomplete so that the N2O escapes from the particle. And then we can, in a kind of crude way, extend our model result here to predict how much nitrous oxide production we would expect in well oxygenated waters. And this is the final result I'm going to show here, again shown in a yield curve form. So these are just that calculation, assuming different fractions for N2O leakage from the particle. So this would just be the fraction of N2O that leaks out of the particle before it can be denitrified all the way to N2. And if you assume somewhere between 0.5 and 2% of all of the N2O escapes the particle before complete denitrification, you can pretty well explain this region of elevated nitrous oxide yield at oxygen concentrations going up to 150 micromolar oxygen. So my kind of final conclusion there, still a little incomplete, still a little hazy, but we think that some combination of this physical heterogeneity and these particle microenvironments can likely explain the elevated nitrous oxide yield we see, and reconcile the biological view, the incubation experiments, and the geochemical view, what we learned from the large scale traces. I did want to say something. I can say my thing about future climate in 1 minute, which is now that we've proved that nitrous oxide production really does happen in well oxygenated waters, as the ocean deoxygenates in the future, we should really expect increased N2O production in the ocean. Right now that we've proved this is not confined just to the anoxic zones that may shrink, but is prevalent throughout a wider region of the ocean as a whole. We did some, again, relatively crude calculations to try and figure out, given predictions of future oxygen distributions, how much might we expect the total marine nitrous oxide source to increase. And our crude calculation is that under an RCP 8.5 business as usual scenario, we think the nitrous oxide source from the ocean could increase somewhere around 10 to 15%. I would talk you through that calculation in more detail, but I'm gonna be mindful of time and leave it there with the conclusions that I already talked through.